About ready to go here. Let's see, most people logged on. We usually get around 45 to 50, so might be a few latecomers. Okay, so it's my pleasure to introduce our speaker today, Dr. Alex Wyatt. Dr. Wyatt is a associate professor in urological sciences at UBC, a senior research scientist at Vancouver Prostate Center, and a senior scientist at BC Cancer. At BC Cancer, Dr. Wyatt is the scientific director of the Clinical Cancer Genomics Program, and in this role, he provides leadership in instituting province-wide cancer genetics and genomics testing programs for patients with cancer in BC. His research goals are to identify associations between genomic and epigenomic alterations and patient outcomes in advanced prostate and bladder cancer, and to translate these findings to clinical biomarkers. His team has developed laboratory and computational techniques to characterize these features in plasma circulating tumor DNA or ctDNA. And through the applications of these methods to clinical trial cohorts, his teams has demonstrated that ctDNA is highly representative of metastatic lesions that features represented in ctDNA can predict targeted treatment resistance, and that prostate and bladder cancer continue to rapidly evolve across lines of systemic therapy. Nationally, Dr. Wyatt is the chair of the Correlative Sciences and Tumor Biobanking for the Canadian Cancer Trials Group, and through his role is involved in the design and execution of phase 1 to 3 clinical trial protocols across Canada. So look forward to hearing more about what Alex has to say today, and thanks again for doing this.
Thanks, Will, and those of you that were involved in my invitation, thank you very much. I'm going to share my screen. One sec. Great. So give me a shout if it's not showing up at full screen, but I assume it is. So I think those of you that do know me know that I'm a genomics researcher. So as Will mentioned, I'm appointed at UBC and the Vancouver Prostate Center and BC Cancer, and I have a large group that does cancer genomics, particularly in prostate and bladder cancer. And we try to work on clinical trials in particular and correlatives around clinical trials. But last summer, had many conversations with the leadership at BC Cancer and also with Stephen Yip and Shah Appleby at the Clinical Genomics Lab, and ended up coming on board in a part-time position as scientific director of the BC Cancer Clinical Genomics Lab, with a kind of goal in mind to help out as much as I could with providing scientific input, particularly around like new assays and bioinformatic aspects of what needs to be done in future. And what I want to talk about today is kind of a roadmap that we're beginning to sketch out. And when I say we, it's a very broad group of people that have contributed so far and lots of different ideas around how we can move towards kind of, I guess, an updated, refreshed genomic strategy that allows us to test more patients in BC with cancer and with better tests. So with that, I'll just start off by acknowledging that we're all joining today from the unceded, traditional and ancestral territories of the Musqueam, Squamish and Tsleil-Waututh nations who've lived here since time immemorial, and we express gratitude and respect for the peoples, cultures and traditions that have and continue to steward these lands throughout generations. So I actually want to start off today by talking a little bit about my own research, just because I think it gives a perspective of one of the areas where I want clinical genomics, certainly within BC Cancer and ideally within our province, to evolve towards. That would just be 5 to 10 minutes. And then I want to talk about what's been the majority of my role with CGL over the last few months, which has really been engagement to try to understand what is the current state, where are all the pain points, where do we have opportunities, and maybe where people are at loggerheads. And then I want to talk about how we're trying to address all of that, the kind of issues that come up through engagement. So both the short-term goals, what we're trying to address right now, and then the kind of bigger vision of the cancer genomics strategy plan. And then hopefully we'll have some time at the end for questions. So to start with my own research, so as Will kind of alluded to, I try to work on, I guess, early biomarker development and then beginning of translating those biomarkers to practice. We study thousands of patients, tissue and blood samples. We tend to generate our own data in-house, and then a large bioinformatic and clinical team that interprets that data and places it in context of outcomes. And so we've helped run multiple prospective trials, and we're involved in correlative efforts around many different international trials. And I think where we perhaps have some unique aspects within our group is we're very disease focused. So we recognize that sometimes you can't abstract the complexity of a specific disease away. And so, for example, if you transplanted me to lung cancer and asked me to give deep insight, I would really struggle, I would imagine. I think that's where my team has really been able to help to have that kind of quite narrow niche. And the other area that's something I'm very proud of is we're really focused to try to engage and help clinical partners both within Canada and around the world. And so we've partnered with a variety of different physicians, whether that's in oncology, pathology, genetics, urology, nuclear medicine more recently, to kind of gain access to different trials around the world that have collected samples that we would be interested in studying. And so that really helps us, I think, stay on top of the latest emerging trends in GU cancers in general. And I'm very grateful to everybody on this slide. We particularly focused on cell-free circulating tumor DNA, and many of you will know that we've all got cell-free DNA in our blood. It's released into the bloodstream by dying cells, and most of what's in the blood is coming from your white blood cells, your hematopoietic lineage. But in somebody with cancer, a small proportion, although sometimes a large proportion of that is coming from their cancer. And we call that ctDNA, circulating tumor DNA. And it's always, when we take total cell-free DNA, a mix of normal and tumor DNA, just like if you'd sample a tissue. The difference with tissue DNA is the cells, the cancer cells, and the blood cells have already chopped up the DNA for us. So that apoptotic machinery has worked to fragment that DNA, which actually is kind of a really cool property of cell-free DNA, because it means you can actually assess chromatin features from the fragmentation patterns. And that's not something I'll talk about today, but there are ways that you can actually infer, to some extent, cell phenotype from the way that the cell-free DNA is fragmented. And I'd say over the last few years, particularly for cancer genotyping, ctDNA has come to the fore as a kind of a reflex approach for assessing actionable alterations in patients with metastatic disease. So does my prostate cancer patient have a BRCA2 mutation? Does my lung cancer patient have an EGFR mutation? Does my breast cancer patient have a PTEN alteration? These types of questions that are typically addressed with tissue can, to some extent, be addressed with ctDNA, although it's very CT fraction dependent. In what I mean by that is that there are some patients with very low levels of ctDNA, and you essentially have an inconclusive result when you test those patients. One thing that I think is important to remember when we think about ctDNA is not always just as simple as normal and cancer DNA. And I like to point out that, in your normal DNA, you've got cell-free DNA coming from metastasis-induced organ damage. We sometimes see that, particularly in patients with really big invasion of their lungs or liver or GI tract. You'll see the footprint of that DNA in the blood. And you also have big expansions in your white blood cell population, particularly in an elderly population. So we typically call this clonal hematopoiesis if it's in the absence of any other blood phenotype or evidence of malignancy. And in an elderly population, you can also have other somatic expansions in the blood. So, manifesting in the blood, so potentially representing subclinical cancers. So you can't always abstract these away totally, but you can at least sequence the white blood cell DNA in parallel so that you can subtract the patient's germline and all of those clonal hematopoiesis variants. And then you need to use sort of an informed approach when you're kind of an expertise-informed approach when you're evaluating potential ctDNA. So if we were looking at prostate cancer and we saw an ETS fusion, an SPOT mutation, and an androgen receptor amplification, it's pretty difficult to imagine that's anything other than prostate cancer. And you can use that together with your fragmentation profile to have a high confidence that you're studying the person's main clinical cancer. So how can ctDNA tests help manage cancer? I think there's kind of three different areas where they're really making a breakthrough in the clinic at the moment. So one is in the context of detecting cancer. So whether that's in a early setting of sort of people at risk of cancer. So maybe they have cancer predisposition syndromes and you could screen them for the presence of ctDNA. I think mostly where this is making breakthroughs is actually in the detection of recurrent disease. So if you have patients with potentially localized disease and you have local intervention and now you see circulating tumor DNA in the blood afterwards, that's telling you that perhaps not all the cancer was eliminated. So I think, that's one area. The second area is in circulating tumor DNA quantification. So the person has cancer, but the amount of tumor DNA that you have actually gives you a pretty good idea of how aggressive their disease is, with patients that have high ctDNA tending to relate to high proliferative capacity of that burden of that disease and bad biology in general. And they do much worse than patients that have low levels of ctDNA and maybe have more indolent cancers. And similarly, when you start treatment, start systemic therapy in a person with high ctDNA and that the amount is reduced, that can happen really early on in the first few days of treatment, and it's a very powerful potential response biomarker. And actually, I've been working with the RESIST group to evaluate the next steps for incorporating early on treatment ctDNA measurements into RESIST criteria. Because I think that offers really important opportunities for clinical trial design and getting evidence of efficacy in phase 1 and 2 trials really early on without waiting till 3 months into treatment to recognize that a patient wasn't benefiting from a treatment and therefore exposing them to all this toxicity. The final thing I'll mention about ctDNA before I move on to the next part of my talk is that I think where people, particularly in oncology, are most familiar is in the context of characterizing ctDNA that's present. So can you find mutations that are linked with potential drugs? So a BRCA2 mutation and a PARP inhibitor, or a PI3 kinase alteration and a AKT inhibitor, for example. But there's more opportunities than just that, and I think what will be coming in the next few years is sort of where tissue profiling is now. So really getting the whole gamut of alterations that can be present. So not just mutations, but structural rearrangements as well. And then getting at that epigenomic information that I also mentioned. I think that's going to be a whole new wave of biology that we're able to interrogate in late stage patients, that particularly in those where we couldn't get tissue easily before, is going to open up some really exciting new areas for biomarker discovery. So I think just to highlight, this is not just kind of a promise for the future, because a few years ago, CGL, working together with BC Cancer GU group, genitourinary cancer group, actually translated a prostate ctDNA test that is now available in the context where tissue testing doesn't work. So patients with metastatic prostate cancer have tissue testing and concurrent germline testing done, and those individuals for which tissue testing fails or where there isn't a sample available, they can have reflex testing using ctDNA. Now, this is a relatively old generation test, and there's probably a lot of improvements we could do to it, and it's not used on a huge number of patients, but I think it's proof of principle that we can translate this type of thing and make it available to patients within British Columbia. So I think that this is a really exciting proof of concept that we have the opportunity to build on now. So with this kind of like research background in mind, then what I've been doing over the last few months is talking with people around the province and getting a lot of feedback as well from inside CGL and the Genome Sciences Centre and other people involved in delivering clinical genomics care in BC to understand where are the pain points, and where are the sort of deficiencies that need more funding and more people, and I think how can we sort of build on those ideas to fix the problems. So I want to start before I give a little bit of that feedback of where the opportunities are in BC and kind of or at least what is this challenge we're trying to solve. So when we think about precision oncology at the clinical level in British Columbia, I think this top point that I've highlighted in blue is mostly historically where we tend to focus, right? So we think about precision oncology kind of in the context of companion diagnostics. A drug comes along, approved by Health Canada, reimbursed by the province, let's say, and now you want to, but it's only relevant for a subset of patients that you need genomic testing to identify. And that's kind of where there's typically historically been funding to stand up a test that would screen the broader population, identify those patients that would be eligible for the approved treatment. Now, I would argue there's all these other areas where precision oncology is really poised to deliver on improved care that we historically in BC and probably in Canada in general haven't had the funding or the infrastructure to deliver on. So, of course, if pathologists have genomic tools in their hands, there's many instances where they can have more accuracy in their initial diagnostics for certain kidney cancers or lung cancers, for example, where I think having information on fusion gene status or underlying mutation to tumor suppressor genes is going to help diagnostics. Similarly, we know that understanding really aggressive genomic features in the late disease and perhaps the very high-risk localized disease does allow you to more accurately prognosticate. And while this, prognostic biomarkers are obviously not as sexy as predictive biomarkers, they still can inform patient management and, for example, the frequency with which you see patients or might do imaging. And I kind of mentioned the potential for blood samples to help you understand treatment response. And I think this is a really exciting area for potential savings in terms of patient exposure to unnecessary toxic therapy. If we can find out early on that you're not benefiting from a treatment, we can spare the toxicity and we can spare the financial toxicity. So I think that's it's a really important area that's not going to be addressed by pharmaceutical company trials, for example. And then we also know that just having the ability to do broad clinical genomic tests is often now required for us to open clinical trials. And there's probably an increasing number of trials that we at BC Cancer and within our province are unable to operate or open because we don't have the type of test that may be required to run those trials. So ultimately, I think effective genomic testing through precision oncology means that more patients get screened earlier, get the specific diagnosis, receive targeted treatments, reduce unnecessary treatments, and expedite their time to treatment. And I would argue that ideally, what we need to do is move from this blue point at the top down through some of these other points as well. So what are the challenges? And obviously, I don't say this flippantly. We can say that the ideal vision, and obviously where we are is perhaps a long way from that. One of the key challenges is that right now it relies on specialized laboratory testing, and I don't really see this changing in the near term. So you need highly trained individuals, and I would argue with a cancer focus and clinical experience, and often I think you need subspecialized cancer focuses within that. So you can't just take all these broad cancer genomic people. You need people that really understand the individual disease and tumor sites. And you need that close integration with clinical teams and care delivery. And I highlight these 2 points in blue because that's not always that easy. Those are perhaps the most difficult parts to stand up because that requires trained people and people that are integrated within a healthcare system. Additionally, cancer genomics is really rapidly evolving. Constant technological advances always new treatments and genomic biomarkers, and always urgency when it comes to translation. You'll never hear a kind of new drug being approved and then the messaging being, well, we can worry about moving this into clinic in a few years' time. It always has to happen now. We need to anticipate that better, of course. But there's also this need for constant process improvements, education, engagement. So this is a beast that needs the mechanism and the people and the resources to address all these constantly changing variables. So where does the CGL fit in? So the BC Cancer Clinical Genetics and Genomics Laboratory, which we term CGL, is mandated to provide molecular genetic diagnostics and paraffin fish testing for all patients in BC. And they also provide cytogenetic and fish testing for PHSA, Northern and Interior Health Authority. And it's stocked with very specialized personnel with deep experience. You may know that the medical director is Stephen Yip. There's an operations director, Shar Appleby, and there's several clinical genetics leaders that you'll probably have met over the years. Tracy Tucker, Sean Young, Ian Baudet, Bahare, and a new team member, Peter, as well as people involved in optimization and development of tests like Curtis. So these are specialized individuals with lots of training and experience. Further to that, the lab itself is accredited. The team members are accredited as well, including the technologists. And collectively, what they do is not just generate data and form reports, but provide informed curation and interpretation of results. And again, I would argue this needs that bespoke knowledge of each indication and integration with physicians and healthcare professionals around us. And the CGL is really, it's not supposed to be a silo. It should be working within our healthcare system and network to implement, educate, monitor, and improve. But I would argue that because of understaffing and under-resourcing over the years, the ability, the bandwidth to engage becomes necessarily less because you've really got to focus then on service delivery, and that's where all the hours have to go. So as I mentioned, over the recent months, I've been trying to talk to as many people as possible. In the 1 day a week I'm supposed to be working on this job, and I've tried to summarize the engagement feedback that I've received consistently on this slide. And I just want to run through some of these points because I think they're quite important to understand what we're actually trying to address in the strategic plan. So the first point is that we have this combination of too few patients eligible for testing. For example, to date, you can't get a somatic panel testing done for a patient with metastatic breast cancer, for example, or metastatic bladder cancer. There are relatively outdated germline and somatic genomics tests. I think if we look at other jurisdictions, they're standing up perhaps genome sequencing or very large panels to understand a lot of different genes, and potentially we have focused, we haven't had the ability to evolve to newer panels. So ultimately, what this means is you don't test as many patients, and the patients that you do test, perhaps you don't have all the information that you might like. And so this is clearly a barrier to optimal care and opening new trials. We have probably related to the fact that we can't get enough information. There's consistent feedback that, unsurprisingly, there isn't enough information on reports. And that's because we're not testing as many of the genes or not as many as the events as we might otherwise want to do. And paired with that is quite an inhibitory laboratory information system that relies on a lot of paperwork and is probably not really appropriate for genomics testing. The second blue point I'm highlighting here is really about the lack of kind of a consistent process for how tests change over time. Everything tends to be very ad hoc and knee-jerk in response to a new indication coming online, a new drug coming online. And ultimately what that means is that you have kind of a bit of an undermining in the opinion of transparency around what's happening. and then any decisions to fund or not to fund. And it probably also makes things more difficult to fund because there's no consistent system and sort of reproducible way of doing things. There is, as I touched on this earlier, because of the understaffing, and I would argue the under-resourcing of clinical genomics in the province, there isn't that capacity in the system for continuous engagement. And I think if you talk to anyone at CGL, they want to do it. And likewise, if you talk to anyone with oncology, pathology, laboratories, and so forth, everybody wants to engage, but we have to create that capacity within the system. And you do that by hiring additional people so that every person has a little bit of extra time rather than having to focus every minute of the day on service delivery, for example. The challenge without having this capacity for an extended period of time, which is what we've had, is that you end up with fragmentation across the province and different health authorities starting to think about doing things themselves, not leveraging our combined expertise as a group, inevitably end up looking inwards. And then lastly, there's probably inefficiencies in the way that samples are processed. We could do with more automation. We don't have the resources or the capacity for process improvement. And I would say, the we is very broad there. I think it's probably the case across diagnostics in general. And then there's some physical issues. There's lack of space and infrastructure, compute power, and far insufficient number of people. And lastly, there's no capacity or framework for clinical genomics research. So, we obviously have some great genomics research infrastructure within our province. Many top researchers, both at BC Cancer, VCH, and UBC, and of course the Genome Sciences Centre. But what we haven't really been able to tap into over the years is using the results of clinical testing to improve our research. And so that's true both for sort of just physicians wanting to look at the aggregate of mutations that may have been present in their patients over a year and trying to understand how that may or may not have contributed to care improvements. But it's also for sort of quality assurance as well and research into that. So I think that's really something that I think many people in our community are crying out for, and again, not been possible to date because of lack of resourcing. So what's the way forward? Well, I think there's two opportunities here. One is short-term kind of improvements, leveraging some of the Cancer Action Plan initiative that Kim Chi has spearheaded, and sort of using some one-time funding to improve the tests that we may have available and have a little bit of extra capacity in terms of personnel. The longer term strategy is probably more important because that's about securing bigger operational budgets so that we can sustain more people working in clinical cancer genomics, both within BC Cancer and then engaging outwards across the province. And I want to go through each of these two areas now. So, the first main bullet point here is about the short-term goals, and this is where we can leverage some of the existing Cancer Action Plan funding to address some of these deficiencies. So, can we upgrade some of the assays and workflows? Potentially, can we expand a bit of our dry lab space and recruit some extra specialists? So whether that's medical geneticists, some bioinformaticians, and so forth. And really, I would argue this isn't even about expanding what we're doing. This is about meeting current demands and case volumes, and also enabling the partners that we're trying to work with. For example, the Genome Sciences Centre with whom we partner, I think would really benefit if we had increased capacity to interact with them and share some of the burden and be more able to sort of direct their operations to some extent. And ultimately, this is really about delivering what I would say is the current standard of care. So, what are those challenges? So the CGL, as I've kind of been trying to emphasize, is understaffed and under-resourced. And as many of you will be aware at PHSA, the new hire process is very slow. So we've been trying to bring on people since I started in August and potentially before that. And the approval process is is lengthy, and I still have not been able to hire anybody myself, despite having funding available. And hopefully that will change in the coming months. But that's a kind of frustrating reality of our jobs, of course. We're at the limit of our physical space, and we also haven't had kind of investment in computational resource or bioinformatics capacity. I've touched on these other points on previous slide. I think I just highlight the last points here because talking at pathology rounds, my sense is from talking to various pathologists, both within our local network and also externally, I think there has been some, I would say, disenfranchising of pathology. I think that's partly because of the under-resourcing, right? But I think the central genomics model is not always preferred, and I completely understand that. There's some that are perhaps perfectly capable to stand up genomics testing in their own jurisdictions. But I think what's key is that pathologists are clearly a very valuable resource in the province that could help a lot with clinical genomics, especially if we start to try to move things towards diagnostics as much as it is about currently companion diagnostics. And probably there's mechanisms to better include pathology within clinical genomics workflows. So where are we going with, I don't want to just sound pessimistic and moan about stuff. So I think we do have some cool things coming, and some of those I just want to highlight on this slide. So we are working, and when I say we, I'm really not talking about myself. These are initiatives that others within the group have been working on for a while, and I get to kind of just come in and cheerlead a little bit. But one of them is that for many years, as you may know, the Hereditary Cancer Program has partnered with CGL and GSC to do targeted sequencing on the germline of people diagnosed with certain types of breast, ovarian, prostate, and so forth cancers to look for cancer predisposition variants such as in BRCA2 or mismatch repair genes. So excitingly, we're actually about to be able to go live with swapping this test for a whole genome, so that patients that are indicated for hereditary cancer germline testing would actually have a whole genome generated. And then the clinical report would provide information on a core set of cancer genes. But that whole genome backbone would mean that we have the capacity over years to then go back and refine what we do and potentially even have different virtual panels depending on the cancer or the indication. And of course, I'm sure you can see that that type of underlying data would also be really valuable for research over time. So similar to the germline, we're trying to upgrade on the somatic testing as well. And what we're hoping to do this year is replace the kind of the older targeted sequencing approach that looked at less than 100 genes with a newer approach that covers about 500 genes. And the idea is that initially we would learn to walk before we can run, and we will be reporting out on a smaller number of those genes. But as time goes on, we can grow the number of genes that are reported on from this panel, including higher order metrics like tumor mutational burden, microsatellite instability, whole genome doubling status, and so forth. There's also, also planned is to at the same time to move from the kind of older myeloid targeted panels to a similarly increased size solution for blood-based cancers, and also to build upon the work from the lymphoma team working with GSC and CGL over many years to have a clinical lymphoma panel as well. So, these panels are about creating capacity for expanded testing. They're not about doing expanded testing yet, because this doesn't then suddenly mean we can take a patient with bladder cancer and test them, because we won't have the funding for that. But I really think this is the necessary first steps to be able to offer expanded testing and improved service. I think importantly, it's going to build on the existing BC Cancer and provincial infrastructure and networks. So it uses all the partners that we're already working with, like Genome Sciences Centre, for example. And so I think it's a really exciting suite of changes that we have coming over the next year or so. So what about the bigger picture, which is, well, how do we increase long-term operational funding And why would we do that? Well, it's even just to meet expected case volume increases without even thinking about extra cases. But ideally, we also want to expand testing to urgently needed areas, so that we're not just testing some cancers, but we're testing as many cancers as possible. And then I would argue that this is really just needed to deliver on clinician and patient expectations for precision oncology at the clinical level. So what we've been putting together in draft format so far, and why I'm kind of doing this like roadshow to get feedback, is to create an integrated strategic plan for a flexible centralized platform for real-time genetic and genomic testing in BC. And so this would both improve precision oncology, as I've been mentioning about, but also give opportunities for research going forward. And what we would advocate for, what we want to advocate for, is germline testing eventually for all people diagnosed with cancer. So that obviously requires quite a long ramp up. But this is a vision that Intan and Sophie and many at the hereditary cancer program have been really pushing for years. And I want to be able to help with that. And I'm sure many of you also agree. Also, somatic tissue testing for all patients with metastatic cancers, where there is the possibility of clinical trial enrollment and actionable alterations that could influence systemic care, systemic treatments. But also, there's guideline endorsed localized disease testing in some cancers. And then you won't be surprised to learn that I'd also really like us to have a plasma ctDNA strategy for genotyping in patients that don't have tissue available, or where the tissue is no longer representative of their disease. And ultimately, I would also hope to be able to evolve that so that we have some assays that could be used for clinical trials of those kind of patient-centric questions, like dose de-escalations or reducing of toxicities. So one of the questions I've had, very fair question, is like, why are we drafting this plan? Is this BC Cancer's strategic plan, or is this BC's cancer genomic strategic plan, right? And I mean, it has to be a bigger vision than just about BC Cancer. We happen right now to have the mandate and the resources through the 10-year action plan to kind of kick things off. But ultimately, we, meaning BC Cancer, serves the public, not just those within BC Cancer. And I think at the minimum, what we have the opportunity to do is determine good practices. And so, we can kind of provide a model for that. We don't have to force everybody to adopt it, but I think we can lead the way in what we believe those best practices to be. We're going to continue, especially once we have hired some people to engage with many stakeholders as possible, and to try to incorporate suggestions as we go. So I don't really see this as being some fixed program that a few months of chatting to people kind of then we shut our ears off. The idea is this should be constantly evolving and learning, and we will build in the mechanism for constant process improvement. I think the last point here, we do need to think about anything we build. How can that help areas beyond genomics? So I mean, even CGL does much more than clinical large panel genomics testing, right? So things like karyotyping and cytogenetics, and in some cases single gene testing and so forth. But I'm also beyond that. How can other diagnostic labs or imaging-based setups benefit from any infrastructure that we're looking at implementing, like laboratory information systems and so forth? So what are the key features of the plan? So the first is to establish enablers to deliver cancer genomics testing within BC Cancer. So things like, how does the governance of CGL need to change or be adjusted? Where do additional people need to be fitted in? So technologists, informaticians, and so forth. To build that capacity for engagement and outreach to oncology, pathology, partners, and so forth. Mechanisms for new test development, and processes and terms of reference for any changes that we make. Improving the way that we report results, so having actual databasing systems for that, having improved kind of data-driven reports and a degree of automation in them, so that to kind of reduce the burden on time-pressured people at CGL. Partnering with the health system to enhance, I should say enhance, not enhancer, enhance supports. So things like laboratory information systems, data management systems, and so forth. There are some things we obviously can't control, but we can try to influence, what should the perfect recipe be for a laboratory information system that works well for genomics. We need to clearly improve automation, streamline systems, provide mechanisms for sample tracking once a test has been requested, optimize turnaround times, have mechanisms for better quality control and quality assurance over time for the oncologists and pathologists and surgeons who are ordering these tests. So really generally about how do we improve efficiency for clinical users. Clearly, we need funding so that we can increase the space and the footprint. So more people need to go somewhere. So that includes a lot of dry lab space and office space, data management and compute solutions, and mechanisms for people to access data as well. And lastly, we need a platform for advancing research. And other big academic institutions around the world have implemented some degree or another of universal consent. And I think this is a worthy goal that we could build towards at BC Cancer, where patients having clinical genomics testing are offered essentially the opportunity to consent into a research program. So I think that would then enable us as a community to do a lot of work with the data that's being generated. But beyond that, we also need improved databasing. We need to be able to run clinical trials, and we need to be able to do research, at least at the aggregate level, on the data that we've generated. There are some soft goals as well. I recognizing this has been a lot of text. I've only got one more slide after this, and then we'll stop. But I appreciate, and if anyone's still listening, I don't actually know because I can't see anything other than my own screen. But I appreciate you holding on. So soft goals, I think we have to increase engagement. I mentioned this word many, many times, but that's clearly a deficit at the moment. I think we need to be flexible for the exact mechanism that we implement genomics testing, right? I think many of us within CGL and myself included, and don't want to speak for Stephen yet, but I think he would probably also advocate for this too. We'd prefer the central model of testing because of the specialized nature of the lab setups. But I think that's not preferred by everybody, and we don't want to be always the bad people. And I think so. What we have here is the opportunity to set standards and even assays for others, right? We can leverage existing partners and infrastructure where possible. We don't need to reinvent the wheel in some areas. We don't need to be potentially going out and buying a ton of Illumina NovaSeqs when we have someone 3 blocks away that has those. Similarly, we may not need to hire a bunch of molecular pathologists to help with report sign-ups when we have those people within our community. The kind of penultimate soft goal is how do we increase transparency and formal processes in general. An obvious example of this is to make sure that we have specific times of year when you can request new tests or upgrades to existing tests, and that becomes known, just like it is for PEC approvals, for example. But there's also opportunities to create transparency in terms of the way that we provide information about where a sample is in the pipeline, and in terms of the types of information that we're providing back in terms of genomic results to the end users. And lastly, I think we all want better quality assurance of clinical genomics. It's been difficult with the current way that databasing is set up to say, well, to what extent has having these genomic tests improved cancer care? And I think the onus is on us to prove that over time. And so that will be a key soft goal for me. Oh, I missed one. I have touched on this already. Allow BC to operate those patient-centric trials, as I touched on. So this is what the draft roadmap looks like for the strategic plan. And I would emphasize this is kind of, being exposed to feedback and ideas right now. So this is by no means final at all. But these different colors are representing those different points that I mentioned. So you can see establish the enablers, improve test reporting. Those are the different categories that I mentioned. And our idea is to sketch out how we might try to address these over a 5-year period. And this has really been done in concert with the BC Cancer Strategic Planning Implementation Team, SPI team. So again, I just get to be the cheerleader and kind of talk about this, but they've really done a lot of thinking behind the scenes on how this could work, when we would need specific people brought on board, who we would need to engage with at each time point. But I think, recognizing that there's only a few minutes left in the session, what I would like to do, maybe I can ask Will or others to circulate these slides afterwards, but we'd be really interested in hearing feedback about this, and what have we not thought about. What do you agree with as well is always nice to hear. But where should we be making adjustments, and who have we left out? So I think with that, I will stop my monologue and pause. And if there's anybody still there when I unshare my screen, I'm happy to answer questions.
Thanks very much, Alex. Still lots of people left online here, so we'll open it up to some questions. It looks like Steve Yip has one, so I'll let Steve go.
Yeah, thanks, Alex. And I think Alex and Shaw has been a godsend to help with this initiative. This is a really big deal. And as I tell people, this is like once in a lifetime opportunity to really improve genomic delivery. And traditionally, pathology has always been underappreciated in medicine. So I think it's time that we need to really improve our delivery, and particularly for precision oncology and efforts such as whole genome sequencing and like pan-cancer liquid biopsy initiative will significantly improve the care of cancer patients, to get them into trials. And lastly, centralized testing is great, but we do need to recognize that we have a lot of colleagues outside that really want to get into the game because of the training, their own interests. So we really need to be collaborative and engage them in how we plan these assays, and also involve them in signing out and helping with the troubleshooting. Or else it's just going to be, a nation state of molecular testers, and everyone's going to have their own machine that would dilute out our ability to offer these tests in a efficient and cost-effective manner.
Any other questions? Oh, go ahead, David.
Hi, Alex. A fantastic talk. This is like a geeky research question, but what can be learned from the qualitative and quantitative analysis of not circulating tumor DNA, but the host-based cell-free DNA? Can you learn things about inflammation status, immune system activity, or perhaps even response or prediction of chemotherapy side effects?
Yeah, that's something we've been super interested in, actually. So particularly the side effect profile, we believe could be anticipated after one cycle or so of treatment, based on basic presence of non-blood cell-free DNA, or indications of non-blood cell-free DNA. I sometimes make the joke that if one of my kids punched me in the liver, that I'd be able to see the effect of that the following day, in the cell-free DNA methylation features. And I don't think that's entirely like a joke, really. We have seen that when patients have certain dominant patterns of metastasis, so let's say we have a prostate cancer patient with particularly large number of lung lesions. The lung methylation profile or fragmentomic profile can actually be more powerful than the prostate one in the blood. So it really means there's a lot of, it's probably related to the fact that the lung progenitor cells are having to go into overtime to replace those that are being damaged by the metastatic growth. So I'm quite optimistic about that. I don't know about, I think it'll be inflammation and changes in kind of immune profiles might be more challenging, or might not, cell-free DNA might not be the best way to go about that, because then you're looking at the background signal of other blood cells, and the confounder is quite high, and maybe then it's the blood cells themselves might help you. But yeah, I think that's a great opportunity.
Okay, thank you.
Jennifer has a question in the chat here. Is there a potential marker on the horizon that may support earlier diagnostics of pancreatic cancer?
Yeah, so I think the most exciting areas for ctDNA to help in early diagnostics is probably in particular at-risk populations at the moment. I think we haven't really addressed at all the value of screening general populations yet for all cancers, right? But what's exciting is that the methylation features on cell-free DNA and ctDNA, or the fragmentation profiles, can be very sensitive for detection of non-blood lineages in circulation. So in theory, if someone has an early stage pancreatic cancer, some of that DNA will be present in the blood, and it could be detected. I think what we don't know yet is the specificity of those signals. And then how do you distinguish between slow growing and fast growing lesions? So, if we had a very sensitive test for prostate cancer, we'd probably see prostate cancer in most old men. But that doesn't mean we would need to then intervene in all of those people. So I think that's, yes, there's a potential solution on the horizon, but I would say that's quite a long way off on the horizon to be thinking about pan-cancer early detection tests.
Yeah, I was wondering something similar with lung cancer. Well, lung cancer, you get high-risk individuals that are undergoing screening, but has there been any tests in that population? It means that seems like a one that you would be able to, correlate with a lot of outcomes pretty rapidly in a pretty defined manner with a lot of metrics like, the CT scans themselves, all that. That would seem to be something that maybe we should discuss at some point, especially considering that, that's just rapidly rolling out in terms of implementation right now across the province as well.
Yeah, I think we should be collecting plasma from these patients who are undergoing low-dose CT screening and breathomics and stuff, particularly in the high-risk, female non-smoker Asian population.
Exactly. And those are the ones that we're genomically characterizing right now, their tumor tissue DNA too. So maybe something we should discuss. I don't know if there's any other questions. The only other one I had is what are the big barriers to universal consent? Like, I mean, you obviously see a lot of these institutions like Sloan Kettering, where I know everybody's undergoing impact sequencing and all that. It's like a blanket kind of like, what are the main barriers to that here?
I think it's actually just operationalizing it.
Is it?
Yeah. So, especially at initial diagnosis, right? There's already a lot being thrown at the patient. How do you operationalize them signing a consent form as well? And, so it would need to be something obviously initially discussed a lot with REB and other stakeholders. But if you did have a path forward, it would have to be integrated within the EMR and Cerner, for example, a BC Cancer, and it would have to be pretty simple, e-consent as well. Yeah, I think it's operational. It's not like really. I think it's a solvable problem, but operational. And I would say it's really where BC Cancer could leap ahead of centers like Memorial, because that's a tertiary referral center, right? So yes, they get all this information, but it's not actually tied to outcomes in a large proportion of the patients that are profiled. It's something we have in BC is the ability to follow patients long term. So.
And more representative population.
And also lastly, we're also working with Dean Regier's group at Health Economics to sort of link genomic features and large data to patient outcome, like linking to different tumor types and time to recur and all that. So his group's actually developing a dashboard within BC Cancer that we can actually track all this data. So I think it'll be really amazing to link genomic molecular testing data with population-based data metrics and where they live. For example, like if they live in Kamloops, like 2 years after a forest fire, is there an
Mhm.
increase in incidence of lung cancer? But are we seeing more EGFR-driven lung cancer versus KRAS?
Very cool. Very cool.
Well, thank you very much for having me, Will, and for the
Yeah,
invite.
thank you very much. I mean, pretty complex issue, but like Steve said, it's pretty exciting at the same time to be part of the leading edge of getting something like this set up. So it's very cool to hear about. So thank you very much again for taking the time.
Yeah, no problem. See you all soon.
Great. Have a good weekend, everyone.